Hello everybody. In this video, I discuss you important topic that is called post-transcriptional modification. Transcription means synthesis of RNA and post-transcriptional modification means the modification of those RNAs. And please understand this process takes place only in eukaryotic cell. When I discuss you the post-transcriptional modification of RNAs which are produced in the eukaryote, I discuss you the post-transcriptional modification of hnRNA to make you mRNA. I discuss you the modification of 45S pre-ribosomal RNA to make you smaller types of ribosomal RNA, that is 5.8S, 18S and 28S types of ribosomal RNA. I discuss you the various modification, extensive modification which takes place on the transfer RNA. So it is not that it is only the hnRNA which is undergoing post-transcriptional modification as to make you mRNA. We also have the modification of ribosomal RNA and transfer RNA under this heading. So first of all, I start with the modification of hnRNA to mRNA, and please understand it's a totally nuclear process. If this I draw it as a nucleus and this as cytosol, and this is the hnRNA if I draw, which is much larger compared to the final product messenger RNA, which is seen to be much smaller. The large size of heteronuclear RNA, hnRNA, heteronuclear RNA or heterogeneous RNA is because of excessive number of introns which are present in it. So when I talk to you about exon and introns, they are alternate arrangements, they are there in the hnRNA, and many of these introns are actually removed during the process of post-transcriptional modification. And once you see the messenger RNA final product, you have majority of it as exons, and of course certain introns towards the end of it may remain intact. The process of this removal of intron and joining of exons together is called splicing, and for this we need to have a complex which is called spliceosome, which I discuss you shortly. So these spliceosome actually are the arrangements which are there on the intron regions, and these are having the snRNA in it, small nuclear RNA in it, which is having phosphodiesterase activity. They have number of proteins in it, and the various factors which are making the body of it. The small nuclear RNA which are there in this spliceosome, they are of U1, U2, U4, U5 and U6 variety. There is no U3, U7, and number of proteins are making the body of spliceosome along with various unidentified factors. And these spliceosome, they remove the introns in a lariat loop structure. They cut it here due to their phosphodiesterase activity, exon intron junction, and this loose intron, which is now hanging loose, folds on itself to make a lariat kind of structure and is getting dislodged from the site. And this is how all the introns are removed in a loop structure form. This process is called splicing, which is certainly going to reduce the size, reduce the length of this hnRNA. This is completely nuclear process, and another modification which is taking place on messenger RNA before it comes out into the cytosol for translation purpose is the capping at 5' end. 5' end capping. And what is this cap? This cap is, please remember, it is 7-methyl guanosine triphosphate capping. It is not 5-methyl guanosine triphosphate capping. It is 7-methyl guanosine triphosphate. As you understand, it's a simply a kind of modified nucleotide which is getting added here. And this kind of capping at 5' end not only protects the messenger RNA from exonuclease activity from 5' end, it also helps in initiation of translation process. The third modification which takes place on hnRNA is the tailing at 3' end, where the poly A tail is attached here. After the transcription is complete, the number of AMP residues are getting added at 3' end, and this is called poly A tail. And for adding this particular tail, we need to have one enzyme that's called polyadenylate polymerase enzyme. What is that enzyme which is adding this tail? Polyadenylate polymerase enzyme. Similarly, when we talk about the cap association, cap binding at 5' end, you must know we have guanylyl transferase enzyme. Guanylyl transferase enzyme, which again is a obviously nuclear enzyme, and this kind of bond which is linking the cap to the 5' end of mRNA is not the phosphodiester 5' 3' phosphodiester bond. Rather, it is a very unusual phosphodiester bond which is called 5' 5' triphosphate linkage, which is adding the cap to the 5' end of mRNA. This kind of modified mRNA then moves out, and poly A tail helps in moving out this mRNA to the cytosol, and once in the cytosol, this mRNA undergo translation. So this is the modification of hnRNA, which is actually preparing the messenger RNA, and that is going to go to the cytosol and enters in the process of translation. So you must know that, and of course it is purely eukaryotic process. In prokaryote, we do not have any of such post-transcriptional modification. The mRNA, as I have told you in the previous lecture, that all the types of RNA are synthesized as a common precursor in prokaryote, and the translation begins as soon as they are synthesized. There is no time for post-transcriptional modification in prokaryote, but in eukaryote we have a proper post-transcriptional modification which is going to utilize a lot of time and energy in the cell. Similar fashion, when I come to the post-transcriptional modification of 45S pre-rRNA, which is produced in the nucleolus by RNA polymerase 1, is cleaved and is making you 5.8S, 18S, 10-8S rRNA, which are going to play a role in making of the ribosome. And the ribosome is the machinery which is involved in translation process. And this kind of modification, which is seen in pre-rRNA, is called cleavage, and this is also actually occurring at the site of nucleolus itself, where it is like getting produced. It's in the nucleolus this post-transcriptional modification is taking place. When I previously discussed you the modification of hnRNA to mRNA, that process was nuclear, but this is a nucleolar process. Coming to transfer RNA. Transfer RNA, as you know, is having a beautiful structure that's got clover leaf appearance, but you can logically understand that it is never synthesized in this fashion from the gene. The transfer RNA is synthesized by RNA polymerase 3, as I have told you in the previous lecture, and that transfer RNA which was initially synthesized was a long linear molecule, huge molecule, was having extra nucleotides towards 5' end and 3' end. So there are many modifications. Number one, as you understand, there is a typical looping in the transfer RNA, which is making you a stem loop appearance, and this is due to complementary base pairs in the transfer RNA, which tend to attract each other and make the hydrogen bonds between them in the stem portion, and that's responsible for making of these stem loop appearance of the transfer RNA. So that's very obvious. Everybody know that the tRNA folds on itself to make you the stem loop appearance or clover leaf appearance. In addition to that looping of the transfer RNA which occurs in the cytosol, you must know various other modification occurs in the cytosol, and number one is the endonuclease activity by RNA polymerase, ribonuclease P. The enzyme is ribonuclease P, which is endonuclease and cleaves around 16 nucleotide long segment towards 5' end. And similarly, we have RNAs D, which is going to trim the exonucleases at 3' end. Then we have certain intron regions also, especially in the anticodon loop, which are getting removed during this post-transcriptional modification. And last but not least, you must understand that we have addition of CCA terminal, to at 3' end. CCA is nothing but it's a sequence of nucleotide which is getting attached at 3' end. We write it like this. ACC, you call it as ACC from 3' end to 5' end, or you may call it as CCA terminal from 5' end to 3' end. And these 3 nucleotides are added later on as a part of post-transcriptional modification of transfer RNA, and they play very crucial role in the translation process. And so this addition of CCA tailing at the 3' end again is opposed to transcriptional modification. And in addition to that, we have modification of bases, base modification. The important example of base modification is conversion of uracil to thymine. You see, RNA is having uracil, doesn't have thymine, but in one of the arm of transfer RNA, we have thymine. That's called pseudo-uridine arm. We have thymine, and this thymine is actually the modified uracil only. Uracil undergo methylation to make you thymine. So this is one modification which takes place. And in addition to that, we have pseudo-uridine, dihydrouridine, methylguanosine, ribothymidine. They all are modifications which are taking place post-transcriptionally. So this in a nutshell is about the post-transcriptional modification of various RNAs in the eukaryotes. Thank you very much.